the first tumor suppressor gene identified and studied was the retinoblastoma gene. Okay? Now, retinoblastoma refers to three things. There's a disease called retinoblastoma, which is a cancer of the eye in newborn infants. Okay? So retinoblastoma is a disease. Retinoblastoma is also the protein they identified causing the disease. And also you've got the retinoblastoma gene. So you've got the gene, the protein, and the disease. So I'll try and not confuse you when I'm talking about retinoblastoma. So what we're looking at here is the disease, okay, the cancer of the eye. And here's just a look at what it looks like. So in newborn infants, you can see this large tumor in the back of the eye, and you can see the tumor in the eye here. And this is an inherited disease. It's familial, and it's being picked up in newborn infants. Okay, so they started to study this disease, and it became apparent over time that the retinoblastoma protein was a tumor suppressor. And when you lose the function of the retinoblastoma, you get a cancer. And for some reason which is not explained, the eyes are particularly sensitive to the absence of this tumor suppressor protein. So during development, if you've got retinoblastoma, then the eyes are the first organ or structure of cells that are sensitive to this protein. Okay. Now there are two types of retinoblastoma disease, and I'll just quickly discuss the two types. The first is unilateral retinoblastoma. So we have two eyes. Unilateral is one eye has the disease. Bilateral is both eyes have the disease. And by looking at the different cases of the occurrence of this cancer in newborn infants, whether it's in one eye or both eyes, and looking at the frequency of occurrence of unilateral versus bilateral, you can then start to make some very good assumptions of what's happening at the genetic level. So some children are born into families with no history of retinoblastoma, and in these cases the cancer is present in one eye. So if there's no familial history, then you get a cancer in one eye. Okay? And if you eliminate the cancer by radiation or removal of the eye, then that individual is at no risk of other cancers in the body. Okay? So it's very clear evidence that this is a sporadic form of the disease, and the mutation has occurred in those cells that develop into the eye, in one eye, and when you remove those cells, there's no risk of other cells forming cancers. Okay? And so this is termed unilateral retinoblastoma, and it's the sporadic form of the disease. Now we have another case of retinoblastoma. So newborn infants in both eyes have this cancer, and there's a family history of this. Okay, and effectively, if you can cure the tumor of the eye, it does not reduce the risk of that individual having other tumors. Okay, so all cells in the body have inherited this weakness, this mutation in the tumor suppressor gene, and that makes the body susceptible to many cancer types. And the first one that becomes apparent is the cancer in the eye of these individuals. So here we have the two types of the disease. We have bilateral retinoblastoma, and we have unilateral. Now with unilateral, the chances of getting additional cancers is very low in these people, even if you cure the disease of the eye. Whereas if it's a bilateral, familially inherited rather than somatic here, then even if you cure the retinoblastoma of the eye, that individual is at high risk of getting other cancers because all cells in the body have inherited the weakness. So how is it then in these individuals that both copies of the retinoblastoma gene were knocked out through somatic mutations? Okay, So because it's such a rare thing to knock out both copies. Okay, so the rate of appearance of the familiar tumors was consistent with a single random event. Okay, and the sporadic tumors, they behaved as if it was two random events. Okay, but the chances of getting both alleles being knocked out through mutation alone was very low. So it was confusing to people as how can you get two random mutations in one cell during development to cause the disease. So here's the two cases. We have the familiar retinoblastoma, which is bilateral, and we have sporadic retinoblastoma, which is unilateral. So in the familiar case, you inherit one mutant allele. Okay, so you require a somatic mutation in the other copy to give the bilateral retinoblastoma. Okay, so early on in development of the embryo, then you pick up this second mutation, which then causes cancer in both eyes. With the sporadic one, you've got the first mutation knocks out the first copy, okay, and the chances of knocking out the second copy is very low, but it does occur. So there has to be a reason, some other mechanisms in the cell that can cause the second copy to be knocked out. Otherwise, it really makes no sense to think of tumor suppressor proteins, because the chances of knocking them both out is so rare. Well, there has to be a mechanism to knock them both out, because as we know now, these things exist. That there's a whole bunch of tumor suppressor proteins have now been characterized, and retinoblastoma was just the first. So how do you solve this dilemma of the very low frequency of knocking out two copies? Well, there has to be another mechanism. So the first somatic mutation occurs at a low frequency, and that gives rise to a heterozygous cell. So you've got a good copy and a bad copy in the same cell. So it turns out that the second good copy is knocked out by a non-mutational event. Okay, and this increases the frequency of occurrence, and therefore we see the disease.